Dongwen Lyu is an assistant professor in the Department of Biochemistry and Structural Biology at UT Health San Antonio. And his current research addresses two broad topics. The first topic is to study the cellular and molecular mechanisms of aging and age-related diseases and discover new cyanolytic drugs, which can selectively eliminate senescent cells to treat aging and age-related diseases. And then the second topic is to study the mechanism of cancers and develop small molecules, including covalent and non-covalent inhibitors and PROTACs degraders to treat cancers. Dongwen published 42 peer-reviewed papers, and his work includes development of first-generation selective BCL-XL PROTAC degrader, DT2216, and second-generation BCL-XL and BCL2 dual degrading PROTAC 753P. DT216 is currently being investigated as a novel anti-cancer agent in phase 1 clinical trial at Dialectic Therapeutics, and most recently he has led an interdisciplinary team to build new assays for target identification, validation, and discovery of new ligase ligands to extend the frontiers of targeted protein degradation. Dongwen, welcome, and we are very much looking forward to your seminar. Well, one thing, I believe you may be muted, or you are mute.
Oh, sorry.
Oh,
so thank you so much for your nice introduction. So today I want to briefly introduce my recent study. We discover a new novel covalent E3 ligase ligand for degrader design. So for the E3 ligase in the PROTAC field, we need more E3 ligase and E3 ligase ligand because we want to generate like tissue-specific or tumor-specific PROTAC. So that's important to to have more E3 ligase and more E3 ligase ligand. So for the PROTAC field, if you already have a E3 ligase, you want to generate more stable or more potent E3 ligase ligand to generate a PROTAC. One way is you can use crystal structure-based design. The other way is you can doing screening to find a new ligand. This is one of the one of the part of the PROTAC study. And the other part is if you identify a new E3 ligase ligand through the a covalent way or through molecular glue, this kind of way, you find some of the compound you can induce protein degradation, but you don't know which E3 ligase those compound can recruit. Then you can use CRISPR screening, use a co-IP assay, use for covalent compound, you can use isotope APPP, this kind of method to identify the E3 ligase. But all the things we need to do is we want to identify more E3 ligase to recruit more E3 ligase ligand to recruit other E3 ligase to generate PROTAC. So natural product actually is a great source for the drug discovery. In the history, actually, aspirin is one of the well-known, very famous drug for from the extract from the willow tree. And in the past 10 years, actually, a lot of natural product on the compound derived from natural product have been approved by FDA to treat different diseases. So pipelogamine, in our study, we found pipelogamine. The pipelogamine is a natural product from the long paper. Pipelogamine has anti-cancer and anti-aging properties based on our study and other people's studies. And in our previous study, we showed that pipelogamine can induce OXR1 degradation in a UPS-dependent manner. It can bind to several different E3 ligases. At that time, and because we performed the study in senescent cells, we are wondering whether PL can be used as a new E3 ligase ligand to generate PROTAC. So before we move forward, we further performed the competitive ABPP assay in the cancer cells, and we found it can also recruit several different E3 ligases. So based on this, we generate the PL PR CDK9 inhibitor conjugates. For the CDK9 inhibitor, we use SS032. Because previously people already use this inhibitor to conjugate with CRBN ligand to generate a very potent CDK9 degrader named SS032. So we generated, we synthesized a series of compounds with different linker lengths and linker types. And we used degradation and cell variability assay to confirm one of the compound named 955 can potently degrade CDK9 and also induce the cell killing. And while the wildhead SS032 cannot induce CDK9 degradation. So based on this, we want to further test whether the warhead or PR or their combination can also, whether those combination can induce CDK9 degradation. Actually, the result is no. Only 955 can induce very nice CDK9 degradation. And later, we want to know because we saw the degradation in the cells, actually, protein can be degraded by several different pathways like proteasome and lysosome or some other pathway. So we first use the proteasome inhibitor like MG32 and another inhibitor, and we demonstrate 955 induced CDK9 degradation is through the proteasome system. And we also tested the autophagy and lysosome pathway. We used the inhibitor to inhibit, and we found those inhibitors cannot block 955 induced CDK9 degradation. And we also tested the caspase cleavage inhibitor, because when you add in the CDK9 inhibitor, sometimes you can cause cell killing. And we want to see whether you block the apoptosis, whether you can block this 955 induced CDK9 degradation. Actually, this inhibitor can also not block the degradation of 955 of CDK9. So based on those results, we confirmed that 955 induced the CDK9 through the proteasome system. And later, we also used the E1 inhibitor and the deletion inhibitor. We demonstrate 955 induced the CDK9 degradation is through the choline-related E3 ligase. So those results confirmed 955 is a real PROTAC. So because PR contains two double common bonds, we want to test whether PR binds to the E3 ligase through the covalent way. So we generated this compound named 336. We removed the two double common bonds and we tested the degradation, and we found only 955 can induce CDK9 degradation, while the modified compound without two double common bonds cannot, induce CDK9 degradation. So with those results, we get to the close that the E3 ligase recruited by 955 or PR is the choline-related E3 ligase, and they bind the E3 ligase through a covalent way. And in this similar time, actually, Daniel Namura group from UC Berkeley, they identified the nimblite from neem tree. When they conjugate nimblite with JQ1 to generate this PROTAC compound named XH2, they found this compound can induce very nice BRD4 degradation. And in their study, they use the isotope ABPP to identify the E3 ligase. And they finally found the E3 ligase is ARF114. Actually, also in the similar time, another paper published by Xiaoyu Zhang from Bone Cravat group, they found a compound, small fragment compound named KB02. When they conjugate this KB02 with the FKBP12 ligand, SLF, they generated this conjugated compound. They found this compound can induce FKBP12 degradation in the nuclear part. Actually, they also used the isotope ABPP to try to identify the E3, but they failed. The reason is because only the PROTAC compound only recruits less than 10% of the DCAM16 to achieve the degradation. So later they try to use the QIP assay to identify the E3 ligase. Use QIP assay, actually they found several E3 ligase, and later they use like a knockdown and knockout assay to confirm DCAF16 is the E3 ligase recruited by this KB02 conjugated compound. So based on those studies, actually, we know we can use like isotope ABPP or use like a QIP assay to identify the E3. But when I think about the like ternary complex formation, because when you the PROTAC recruits the protein of interest and the E3 ligase to form ternary complex, this kind of ternary complex sometimes it's not so stable. So we came across this kind of method named proximity biotin labeling. Proximity biotin labeling compared with the QIP assay, it can detect low affinity at transient protein-protein interaction. So in 2018, actually, Alice Team group published a new generation of the biotin enzyme named TurboID through the directed evolution method. As this TurboID has extremely high affinity and can biotinate proteins in only 10 minutes. So at that time, we are thinking whether we can adapt this TurboID method to identify the E3 recruited by 955. So we fuse our protein of interest named CDK9 with the TurboID. We generate this plasmid and we transfect the cells with this plasmid. When we get the cells, then we divided the cells into two groups. One group we treat the cells with biotin, and the other group we treat the cells with the biotin and 955. Of course, in this process, to block the degradation, we add in MGR32. And after treatment, we collect the sample and extract protein and use strip-tamping to enrich the biotinated proteins. And later, we use the mass spec to identify the E3 ligase. And the result shows that several different E3 ligase can be highly enriched in the 955 treatment group. And interestingly, KEAP1 is the only proteins also identified by the competitive ABPP in our study. So later we use the knockdown and knockout assay to confirm if you knock out or knock down KEAP1, you can block the 955 induced CDK9 degradation. Of course, we also tested several different other E3 ligase, and we found only KEAP1 knockout or knockdown can block the degradation. So to further confirm 955 can help the ternary complex formation and covalently bind to KEAP1. So we used the nanobrite assay because this assay is generated by Promega. And we compared the 955 and the 336 because we know 955 can induce very nice CDK9 degradation, but 336 cannot. So the result shows that only 955 can induce dose-dependent ternary complex formation, but 336 cannot. So later we also use the gel-based ABPP assay to confirm 955 can compete with IM-Alchem probe to bind to the purified KEP1 proteins. And we also use the KEAP1 inhibitor like CDDO-ME, CDDO-IM, and DMF. And those inhibitors, the pre-treatment of those inhibitors can block the 955 induced CDK9 degradation. So those results, with those results, we confirm 955 can bind to KEAP1 to recruit KEAP1 to degrade CDK9. So another question is whether our compound are specific, to CDK9, or then we use the TMT-based proteomics. We treat the cells with lower concentration, it's 0.1 micromolar concentration of 955 for 1 hour. And the other group, we treat the cells with the 955 with 0.1 micromolar for 6 hours. And the third group, we treated the cells with the 10-fold higher dose of the SS032 for 6 hours. And if you look at the profile, you will see 1 hour treatment can induce great CDK9 and CDK10 degradation. And with longer treatment, you can see there are some, besides CDK9 and CDK10, you will see some other protein also decrease. But if you compare the profile between the 955 and the inhibitor, you will see most of those proteins, those blue dot protein, you will see they are overlapped. That means those proteins, they are downstream protein of the CDK9 or CDK10. So later we use the Western blot assay to confirm 955 can also induce CDK10 degradation through the UPS system, and also the E3 ligase is cooling related to E3 ligase. And in a previous study, as I mentioned, the CRBN-based CDK9 PROTAC also showed moderate CDK10 degradation. I think CDK10 might be one of the target protein of the SS032, but the binding affinity might be low. But even lower binding affinity, the PROTAC still can degrade the CDK10. It's a very interesting phenomenon. And later we want to see whether 955, whether it's more potent than SS032 to kill tumor cells. So we performed the RSeq, and the result shows that with longer treatment, like 6-hour treatment, lower dose, we use 955, we use 0.1 micromolar. But for the warhead or the inhibitor, we use 1 micromolar. You see with 10-fold lower dose, it can achieve similar profile at the R level. So then we further check the downstream genes regulated by CDK9, like CMYC and MCL1. We found 955 is about 10-fold more potent than the warhead. So we also further tested 955 in prostate cancer cells. We choose 4 different prostate cancer cells, 2 AR negative and 2 AR positive cells, and we found 955 showed much better cell killing in those cells. And the result shows that 955 can induce protein degradation in one of the prostate cancer cells. Those results confirm 955 is a much more potent anti-tumor agent than the SS032. So then with those results, we think PL can be used as a new covalent E3 ligase ligand to recruit KEAP1 to target proteins and trigger protein degradation. So we want to test whether PL can be used as a ligand to target other protein of interest. So ALK protein is a kinase. These ALK proteins always fuse with EML4 in non-small cell lung cancers. And in those patients, actually, those patients are very resistant to the ALK inhibitor, such as serotinib. So we conjugate serotinib and the ALK inhibitor with PL to generate this conjugated compound named 819. This 819, when we tested in the non-small cell lung cancer, we found it can induce very nice ALK fusion protein degradation, while the warhead cannot degrade EML4 ALK protein. So we also confirmed that those kind of degradation is UPS system dependent. So with those results, we think the PL is a new E3 ligase ligand can be used to target many other protein of interest. So in summary, I want to mention that we generated the PL-based CDK9 degrader, and we found the TurboID-bait assay is a powerful tool to identify KEAP1 as E3 ligase recruited by 955. And more general, we think natural products are an important source for the discovery of new E3 ligase ligand. And the TurboID-bait assay is the powerful tool to identify E3 recruited by natural product or not other novel E3 ligase ligand or even molecular glue. So, of course, there are some questions we need to answer because we need to further optimize the PL compound to make it more specific. And we also need to answer whether those two, which of those two double compounds plays major role, and the binding site of the PR or the KEAP1. So we are still working on that. We are trying to solve a structure to of the KEAP1 CDK9 ternary complex to get more insight and design better KEAP1 recruiter to degrade other proteins. And I want to emphasize the TurboID-bait assay. Currently, we use this assay in our study to identify the E3 ligase recruited by 955. But this method actually can be used to identify some other phosphatase, because recently in this review paper, they showed that you can use chemical way to induce protein modification. So actually, we think this kind of TurboID method, TurboID-bait method, can be used to identify more like compound to generate like a duck tag or false tag or some other different, lots of different tag. So with those, I want to thank my lab, Dr. Zhou's lab and Dr. Guangren Zhen's lab. Jing Pei is a PhD student, and he performed most of the biological study. And Yufeng Xiao, he generated a lot of the new CDK9-based CDK9 PROTAC, and Xin Gui generated the ALK PROTAC, and she also generated the PR probe for our study. And I also want to thank other people in the University of Florida and the University of Texas. And I also want to thank NIH and Massey Cancer Center to support my study. With those, I want to thank your attention, and I'm happy to take questions.